This series features speakers in the early stages of their careers, such as PhD students, postdocs, and other early career researchers who study endocrine disrupting chemicals. This webinar is also hosted by the EDC Strategies Partnership. The partnership members include the Collaborative for Health and the Environment, Healthy Environment and Endocrine Disruptor Strategies, or HEEDS, a program of Environmental Health Sciences, Commonweal's Biomonitoring Resource Center, and Health and Environment Alliance, or HEAL. So we'll disable the chat during the presentation, but please type your questions in the Q&A box at any point, and we'll get to as many comments and questions as we can after the presentation. If you'd like to activate your closed captions, you can do that by clicking the button on the bottom of the screen. This webinar is being recorded, and it's scheduled to last for 1 hour. I'd like to note that the webinar was originally posted as being half an hour in length. So I know some of you may have to leave at the half-hour mark, which is totally okay. So for anybody who's not able to stay on for the full webinar today, please do watch the rest once we post the recording. If you'd like to download the slides and links to the text of the studies that are mentioned in today's presentation, you can find them on the webinar page and our website at healthandenvironment.org. And we'll actually put that link in the chat in just a moment. So yeah, let's get started. I'm particularly excited to welcome you to this webinar. This topic is so interesting and so important. I'm actually conducting my dissertation research on prenatal pesticide exposure and child neurodevelopment. And we suspect, and the research suggests, that effects on pregnancy hormone levels are a mechanism of action. So there's a lot that I, and I'm sure everyone else, can learn from our speakers today. Today's moderator is Sarah Howard of Healthy Environment and Endocrine Disruptor Strategies, or HEEDS. And Sarah, I'll hand it over to you.
Thank you, Haley. Hello, everyone, and welcome. Glad you could join us. So Haley just went over what I was going to say, so I don't have to do it. So like she said, we started this webinar series as an opportunity for early career scientists to present their research. And this is our 9th webinar in the series. We've had a bit of a pause over the last year or two, but would like to get it going again. So if you are an early career researcher and would like to present, please let me know. The recordings of the earlier webinars are available on the website at the link that I think was just posted. Also, we have a newsletter at heeds.org that you could sign up for, and we announce webinars there, this series and other CHE webinars and so forth. So today our topic is endocrine disruptors and hormone levels during pregnancy. We have 2 speakers who will be speaking in tandem today, followed by a question and answer period. And so to ask a question, please put your questions into the Q&A box because we're going to close the chat eventually. Our speakers are Dr. Diana Pacyga and Brad Ryva. Diana is a postdoc in the Department of Epidemiology at UNC. Her research focuses on how exposures during pregnancy, infancy, toddlerhood, and midlife are associated with maternal and child health. She obtained her PhD from the Department of Food Science and Nutrition at Michigan State University, which is also where Brad is now a PhD candidate, although he's in the Department of Pharmacology and Toxicology. His research focuses on EDC exposures from common consumer products and their relationship with health during pregnancy. So with that, I will turn it over to Brad.
Thanks, Sarah, for that introduction, and thanks everyone for logging in to listen to the webinar. Today we will describe our paper on endocrine disruptors and hormones in pregnancy that was recently published in Environment International. And just to provide a little more background beyond my biography, I'm currently a DO PhD dual degree student at MSU, and this project is one component of my PhD dissertation that I'll be defending this July. Then after my defense, I'll be entering the hospital to finish the clinical portion of my medical school. I started working on this project when I joined Dr. Rita Strakowski's group in the Human Nutrition Department, where Diana was already a graduate student. And she and I worked on this project together while she was in her last year at MSU before heading to UNC Chapel Hill for her current position. On this first slide, we're including a QR code if anyone is interested in looking up the paper after our presentation, but I believe the paper will also be shared as a link in the chat and posted online. Here's a broad outline of today's presentation, where we will cover background, objectives, methods, results, conclusions, and some future directions. But first, Diana will cover the background, focusing on gaps in the current research that we attempted to address in this project.
Thanks, Brad. So I'll jump straight into it. Our work focuses on pregnancy because pregnancy is a very important part of the life cycle. And why is it important? Well, first, everyone that is alive was at one point a product of a pregnancy. And second, an aspect that has often been overlooked until recently is that pregnancy is an important part of the life cycle for the pregnant individual. As a result, pregnancy is a sensitive window for both child and maternal health, and perturbations in pregnancy can have lasting consequences. So, for example, studies have found that pregnancy hypertensive disorders, gestational diabetes, inappropriate weight gain, and preterm birth are associated with adverse later life health conditions in the offspring, including cardiovascular disease, osteoporosis, and reproductive and cognitive dysfunction, just to name a few health outcomes. And this is the premise of the developmental origins of health and disease hypothesis, where what happens in the womb can last a lifetime. Recently, there's also evidence that indicates that these same adverse health conditions in pregnancy may put women at increased risk of later life metabolic and cardiovascular diseases. So it's really important to consider pregnancy as a vulnerable time to protect both child and maternal long-term health. And pregnancy is a vulnerable period due to the many physiological changes that occur, including increase in maternal blood volume, increase in respiratory ventilation, digestive changes, and changes in fat deposition. So during pregnancy, there's virtually no organ or system that is unchanged. And these physiologic adaptations are regulated by changes in the levels of critical pregnancy hormones. So this figure here on the left shows the increase in maternal estradiol, progesterone, and testosterone concentrations that occurs across pregnancy. And these are sex steroid hormones that are produced by the ovaries predominantly until the end of the first trimester, when the maternal fetal placental unit takes over as the major hormone producer. Maternal thyroid stimulating hormone, TSH, which is produced by the pituitary, and thyroxine, T4, which is produced by the thyroid, also change across pregnancy, which is shown on the figure on the right. And changes in sex steroid and thyroid hormones support numerous functions in pregnancy, including implantation, placental angiogenesis, immune and metabolic adaptation, and parturition. And so these are all very important for maintaining the pregnancy health, fetal growth, and development. It is also important to note that sex steroid hormones and thyroid hormones do not exist in isolation. So in this figure, we're just showing a simplified regulatory feedback system, and just to orient you on this figure, so we see that thyroid stimulating hormone is produced by the anterior pituitary in purple in response to thyroid releasing hormone produced by the hypothalamus. And so thyroid stimulating hormone stimulates the production of T4 by the thyroid, and sex steroid hormones produced by the placenta control thyroid hormone levels and production. Additionally, thyroid hormones can influence sex steroid hormone synthesis, transport, and elimination. So there's this feedback loop happening. And so this highlights the dynamic nature and coordinated system of hormones during pregnancy. Therefore, when sex steroid and thyroid hormones are disrupted during pregnancy, this can result in adverse pregnancy and birth outcomes, including infertility, miscarriage, gestational hypertension, preterm birth, and low birth weight. And so it's important to identify the risk factors that are associated with altered maternal hormone levels to protect maternal and child health. And one potential modifiable factor that could impact hormones are chemical exposures. And almost all pregnant women are exposed to classes of chemicals found predominantly in food packaging materials, as well as personal care products and cosmetics, which are the chemical classes we are primarily interested in. And so these include classes such as phthalates. So one example is DEHP from food packaging materials. It's a plasticizer generally used in soft plastics. DEP, which is a scent stabilizer used in perfumes and certain personal care products. We are also interested in phenols, such as bisphenol A or BPA, which is found in hard plastic water bottles, the reusable ones, also canned products. Parabens, including methylparaben, which is an antimicrobial agent found in lotions and cosmetics. benzophenone 3, which is used in sunscreens, and dichlorophenols, which are used in deodorizers and pesticides. And this widespread exposure to phthalates and phenols is concerning since numerous human epidemiological studies have identified associations of these chemicals individually with adverse pregnancy health outcomes, including gestational hypertension and gestational diabetes, along with adverse birth outcomes, including increased risk of having a C-section and preterm birth. And these adverse pregnancy and birth outcomes as a result of phthalate and phenol exposure is likely due to the endocrine disrupting properties of these chemicals. So for the remainder of the presentation, we will refer to these chemicals collectively as endocrine disrupting chemicals, or EDCs. Most of what we know about EDCs is from decades of experimental studies at the cellular, tissue, and animal level. In vitro experiments or experiments at the cellular level, these chemicals have been shown to disrupt hormone receptors and enzymes important for hormone signaling. In experimental animal models, these chemicals can act at reproductive tissue level as well as alter circulating hormones. And this could also be true in human pregnant individuals. As a result, numerous epidemiological studies in pregnant individuals have identified associations of individual EDCs with pregnancy hormones. However, the findings are generally mixed with regards to whether a significant association was identified, and also they're mixed regarding the direction of the association. And the direction of the association might differ within and across different classes of EDCs. So just as an example, some studies found that phthalates are associated with decreased sex steroid and thyroid hormone concentrations. Some reported that phthalates are associated with increased levels of these hormones, while others observed no associations of phthalates with sex steroid and thyroid hormones. And similar mixed associations were observed between parabens, triclosan, and bisphenols with these same hormones. However, in addition to the mixed findings, there are also gaps in the literature regarding associations of EDCs with hormones in pregnant individuals. First, pregnant individuals are becoming increasingly exposed to newer chemical replacements. So for example, the well-known phthalate DEHP is being replaced by DiNCH and DEHP in certain plastic products, and pregnant individuals are becoming increasingly exposed to these two chemicals, along with still being exposed to DEHP. In addition, bisphenol A replacements such as BPS and BPF have been introduced as well. And so this highlights the concept of regrettable substitution, where a new chemical is introduced to replace a chemical that has been identified as problematic without adequately understanding the hazard of the new chemical. And as a result, there have been limited studies that have evaluated association of these phthalate and BPA replacements with sex steroid and thyroid hormones. Another important limitation of the literature is the focus on associations of individual chemicals with sex steroid and thyroid hormones. However, we know pregnant individuals are exposed to multiple endocrine disrupting chemicals, not just single chemicals. So in this figure here to the left, we can utilize this to demonstrate the importance of studying chemicals as mixtures. And so the colorful bars in this figure represent the many chemicals individuals are exposed to from many different sources, including toys, cosmetics, indoor air, and food. Individually, these sources of chemical exposures may not cross the threshold of concern, the dotted line, but as a mixture, the combined risk could cross the threshold of concern, which is this dark bar. And so this highlights the importance of evaluating all these chemicals and these different exposure sources together. However, it may still be important to know the main source of exposure contributing to the health concerns within the context of the mixture. So as a hypothetical example, using the same figure, we know that children play with toys, that's one source of exposure, and they play inside, which is another source of exposure. But it is challenging to determine how these correlated co-exposures, toys or indoor air, contribute to health concerns independent of the other. However, with newer statistical methods, we can say that toys are contributing to health concerns independent of indoor air exposure during playtime and vice versa. And we can also determine which exposure source contributes more towards health concerns independent of the other. Therefore, it's important to understand both the cumulative effect, how these chemicals together impact pregnancy health or hormone levels in our case, and which chemicals are bad actors, the individual contaminants that contribute the most to disrupting hormone levels, accounting for all the related co-exposures. However, there are only a few studies that have assessed associations of an EDC mixture with pregnancy hormones. Importantly, no studies have assessed association of a mixture of EDCs with sex steroid hormones. And the few studies that have assessed associations of an EDC mixture with thyroid hormones reported either inverse or null findings. However, major limitations of these studies include focusing on single-class mixtures. So for example, modeling a mixture of 5 phenols, but not including any phthalates that might come from the same exposure sources. And additionally, a few studies were also able to evaluate replacement chemicals in the mixture. Also, some studies in pregnant individuals, or I would rather say most studies in pregnant individuals, have also typically focused on evaluating linear relationships between EDCs and hormones. However, focusing on linear relationships may result in missing important findings. And so these figures demonstrate just a few examples of nonlinear dose response curves. So for example, in panel B, the figure here shows an increase in response at lower doses, but a decrease in response at higher doses, with the largest effect at doses in the middle. So one difficulty in studying EDCs is the presence of potential nonlinear dose response curves, such as low dose responses that have been identified in vitro and in vivo experiments for many EDCs, including phthalates and phenols. So for example, experimental studies have found that BPA and DHP both display nonlinear relationships with receptor expression, enzyme activity, and reproductive outcomes, including fertility and pubertal timing. And many of the possible causes of nonlinear effects have been proposed, including cytotoxicity, downregulation of receptors, and endocrine feedback loops. And the potential nonlinear associations between EDCs and hormones needs to be investigated in pregnant individuals. And finally, given that maternal sex steroid hormones are produced by the placenta, which is fetal derived, and that thyroid hormone levels have been shown to differ in women carrying females versus males, it is important to evaluate whether associations of EDCs with hormones differ by fetal sex. And only a few prior epidemiological studies have evaluated fetal sex-specific associations. So just to go through a couple examples, in one study, MEP, a metabolite from the phthalate DEP, and Brad will go into much more detail of how we measure these chemicals in humans. But in this study, this chemical was associated with lower testosterone in women carrying females, but associated with higher testosterone in women carrying males. In another example, a study looked at a mixture of 5 bisphenols and found that in women carrying females that the mixture was associated with higher free triiodothyronine, or T3, which is another thyroid hormone, but lower free T3 in women carrying males. And so this is important since numerous prior experimental and human epidemiologicals have shown sex-specific effects of EDCs on health outcomes. And the sex-specific effects of EDCs on hormones could explain these results. So given these gaps, the overarching goal of our study was to evaluate associations of a multi-class mixture of non-persistent EDCs and their replacements with hormones overall and by fetal sex. And this was addressed with 3 objectives, where in the first objective, we wanted to evaluate the overall association of the EDC mixture with hormones. In the second objective, we wanted to identify the major chemicals contributing to the overall mixture association. And in our third objective, we wanted to determine if the association of the EDC mixture with hormones was nonlinear. And with this background, Brad will take you through the methods and the results of this study.
Thanks, Diana, for providing the background. And now that you all have an idea of the background and our study's objectives, I will discuss the methods we use to address these objectives. Addressing these important questions about the impact of environmental exposures during pregnancy is a massive undertaking. And because of the numerous resources required, both the National Institute for Environmental Health Sciences, NIEHS, and the Environmental Protection Agency, EPA, supported and funded a program called the Children's Environmental Health Research Centers, And these centers had the goal of studying the role of environment in many health outcomes, including birth outcomes and reproductive outcomes. And they were primarily interested in air pollutants, trace metals, pesticides, and endocrine disruptors as exposures of interest. Many of these children's centers, along with other individual pregnancy and birth cohorts, joined the Environmental Influences on Child Health Outcomes ECHO study. ECHO expanded upon the scope of the research centers to include numerous factors that impact child health, including various environmental, social, and lifestyle exposures. And the map on this left displays many of the cohorts that are part of ECHO now or in the past. And the small green heart here in central Illinois represents a pregnancy cohort that will be highlighted in today's presentation. That specific ECHO cohort, which we use to address our objectives, is the Illinois Kids Development Study, or iKids, which is an ongoing prospective pregnancy and birth cohort of women from Champaign-Urbana, Illinois. iKids follows pregnant women and their children from early pregnancy through childhood, and women in the study needed to be in a low-risk singleton pregnancy. They had to speak English and live within 30 minutes of the University of Illinois campus, and they could not plan on moving before the birth of their child. For this study, we used information from 302 women. One important aspect to always consider with human epidemiologic studies is what the individual cohort of participants looks like. As the women were recruited in the surrounding areas of the University of Illinois, the women were very characteristic of women living in a Midwestern college town. Specifically in iKids, most women were older than 30 years, non-Hispanic white, employed, college educated, and had an annual household income above $60,000. Additionally, most women did not smoke in the first trimester. A little over half had a pre-pregnancy body mass index considered under or normal weight. And the median diet quality, which was assessed by the Alternative Healthy Eating Index, was 55.8 out of 110 points, but the range was quite wide, suggesting varied diet qualities. Lastly, about half of the women in iKids had no prior births. This is the iKids prenatal timeline, where women were recruited at median 13 weeks gestation and followed throughout pregnancy. For this study, our outcome of interest was early second trimester sex steroid and thyroid hormone concentrations that were measured in plasma at median 17 weeks gestation. This coincides with a critical time period during which the placenta becomes the primary producer and regulator of hormones in pregnancy. Hormone concentrations were measured using ELISAs at the University of Michigan Diabetes Research Center. Specifically, we measured 3 critical sex steroid hormones, progesterone, testosterone, and estradiol, and key thyroid hormones, total T4, free T4, and thyroid stimulating hormone, TSH. During the prenatal window of the study to measure phthalates and phenols, women also provided 5 first morning urine samples through gestation, as indicated on this timeline. In case any of you aren't familiar with phthalates and phenols, urine was used to measure concentrations because these chemicals do not persist in the body, but are metabolized and excreted within 24 to 48 hours of exposure. And because of this, urine is the best medium for approximating exposure to both phthalates and phenols. However, because of their short half-lives in the body, urinary phthalate and phenol levels can vary drastically with an individual. This means that someone's levels of these chemicals can vary remarkably within a day, across a week, or across a month. And given this high within-person variability, we physically pooled the 5 urine samples for EDC assessment. And this is a well-validated approach for approximating total pregnancy exposure to phthalates and phenols. Then the pooled urine samples were sent to the Centers for Disease Control for quantification. Specifically, we measured 15 phthalate metabolites, which are listed in the bottom part of the table that I'm moving my cursor over. And those reflect exposure to 8 phthalate parent compounds, which are listed in bold here. Because phthalates are quickly metabolized and excreted in urine, we have to use urinary metabolites to approximate exposure to the phthalate parent compounds that are actually contained in the consumer products. For example, DEHP is used in plastic food contact materials and is rapidly metabolized into these 4 urinary metabolites. Therefore, if we sum the concentration of these metabolites, we can approximate the exposure to DEHP. In contrast, DEP, which is used as a fragrance stabilizer in perfumes, is excreted as one major urinary metabolite, MEP. Therefore, we can use urinary levels of MEP to approximate exposure to DEP. In addition, we measured 4 urinary metabolites to approximate exposure to 2 phthalate replacements, DiNCH and DHTP. These chemicals are found in plastics as replacements of DHP, as Diana said, and importantly, as she also said, exposure to these chemicals is increasing across time. Moving on to the phenols, we measured urinary levels of 11 phenols, and this includes bisphenols such as bisphenol A, BPA, along with common BPA replacements bisphenol S and bisphenol F. We also measured 4 parabens and 4 other phenols. Lastly, we measured triclocarban, which is not considered a phthalate or a phenol. In total, these 31 urinary biomarkers reflect exposure to 22 EDCs. Unsurprisingly, over 90% of women and IKIDs had measurable levels of most urinary EDC biomarkers, suggesting that most women are exposed to almost all of these chemicals. However, IKIDs women had lower levels of exposure to butylparaben, bisphenol F, and triclocarban, which indicates lower levels of exposure to products with these chemicals in them. Importantly, many of the IKIDs EDC concentrations were comparable to levels in women of the same age in the National Health and Nutrition Examination Survey, NHANES. And this is really important to note because, as I said earlier, the IKIDs cohort is mostly non-Hispanic white and of higher socioeconomic status. But knowing that the levels of exposure in IKIDs are not radically different from a nationwide representative sample, that strengthens our ability to generalize our findings. However, there were a few notable exceptions where EDC concentrations in IKIDs differed from NHANES. Specifically, as you can see in this table that I just highlighted, levels of MEP, the DEHP metabolite from scented products like perfumes were lower in IKIDs women. This may suggest that pregnant women and IKIDs are using less fragrant products or are using perfumes that have had DEHP removed. Additionally, women in IKIDs had higher levels of DEHP metabolites compared to NHANES women. And women in IKIDs may be using products that are higher in replacements, which could make sense if these products are catered to more health-conscious women who are trying to reduce their level of exposure to the classic EDCs like DEHP. Now, as Diana said in the introduction, women are not exposed to one single chemical at a time, but rather they are exposed to a mixture of chemicals simultaneously. Therefore, we were interested in modeling EDCs as a mixture rather than individual chemicals. In order to model chemical mixtures, numerous statistical methods have been developed. This figure on the left shows many of these methods that have been introduced on the smaller circles on the outside. Because of the rapid increase in the number of methods, we're unable to utilize them all, and it can be difficult to know which one to choose. The fundamental criterion in choosing a mixture method should be based upon the research question of the study. And this is because the major mixture methods were developed to address specific questions. And that's what the inner circles on this figure are showing. Those are showing the broad categories of questions that were addressed by the various methods on the smaller circles. As Diana said, our objectives were identifying an overall mixture association and major contributors to the mixture for objectives 1 and 2, and identifying nonlinear relationships for objective 3. In this study, we used 2 statistical mixture approaches, weighted quantile sum regression and Bayesian kernel machine regression. We used WQS to address our first 2 objectives, and although BKMR can be used to address all 3 objectives, for the sake of this presentation, we will focus on one of its major strengths, which is evaluating nonlinear relationships. This slide should look familiar from before. So of the 22 EDCs of interest that I spoke about in a few slides ago, we were able to include 19 in the mixture, including 8 phthalates, 2 phthalate replacement compounds, and 9 phenols, all highlighted in blue. Bisphenol F, butylparaben, and triclocarban, which are crossed out here in the table, were excluded because many women did not have measurable levels of these chemicals. However, our mixture still includes many important phthalates and phenols from products pregnant women frequently use, as well as key replacements such as DiNCH, DEHP, and bisphenol S, to which pregnant women are becoming increasingly exposed. Now that we've detailed the methods of our study, we can jump into the results. As a reminder, our first objective was to determine if there was an overall association of an EDC mixture with early to mid-pregnancy hormones in all women, and to investigate differences by fetal sex. I'm going to start by describing the results in all women. We did not identify any relationships between the EDC mixture and progesterone, estradiol, free T4, or total T4. Our main finding was that a 10% increase in the EDC mixture was associated with a 5.65% decrease in testosterone levels. And this is a relatively strong association, as we are only modeling a modest increase in the mixture, 10%, and this is much stronger than what we observed when we evaluated EDCs individually. Also, while testosterone does not increase nearly as much as progesterone or estradiol during pregnancy, testosterone levels during pregnancy are important for normal pregnancy progression and fetal growth. Lastly, we did observe a slight negative relationship of the EDC mixture with TSH, but the TSH results were more meaningful in sex-stratified models, which I will cover next. When evaluating whether associations between the EDC mixture and hormones differed by fetal sex, we identified no sex-specific relationships with progesterone, estradiol, testosterone, or free T4. We did find that a 10% increase in the EDC mixture was associated with a 1.5% increase in total T4 only in women carrying females. Lastly, a 10% increase in the EDC mixture was associated with a 0.18 unit decrease in TSH in women carrying males. As thyroid hormones are important in both pregnancy and fetal development, these perturbations may be associated with adverse pregnancy health. And these results also highlight the importance of studying sex-specific relationships, because although the EDC mixture was not associated with total T4 in all women, it was associated with total T4 in women carrying females. While it may be more intuitive for some of you to assume that associations of EDCs with sex steroid hormones would be more likely to vary by fetal sex rather than the thyroid hormones, there are plausible explanations for sex-specific findings in thyroid hormones, including that circulating thyroid hormone levels do differ depending on whether a woman is carrying a male or female fetus. However, understanding the exact specific mechanisms of sex-specific differences would likely need to be investigated in future experimental studies. Now that we've identified the overall associations between an EDC mixture and early to mid-pregnancy hormones, we wanted to know which chemicals were the major contributors to the mixture association. As I just showed, the EDC mixture is associated with a decrease in testosterone in all women using WQS. In these models, we found that phenols, triclosan, propylparaben, 2,5-dichlorophenol, bisphenol S, and 2,4-dichlorophenol, and the phthalate replacement DEHP are the primary drivers of the negative mixture association. The percentages next to each chemical reflect the relative contribution to the mixture association. So the top chemical driver, triclosan, is contributing 14% to the association between the EDC mixture and testosterone. So to summarize, in general, our mixture is identifying antimicrobial agents like triclosan and propylparaben, as well as replacements like bisphenol S and DEHP as major bad actors. When we compare these results to those from models where we assessed one chemical at a time, we noticed some similarities, but also some differences. One interesting difference is that while triclosan was the primary driver in the mixture, contributing 14%, when it was evaluated individually, the magnitude of the association was smaller than that for the dichlorophenols and propylparaben. This highlights the importance of evaluating chemicals as mixtures, since the relative importance of chemicals may depend on their co-exposure. As a reminder, the EDC mixture was positively associated with total T4 only in women carrying females. And the bad actors identified in that model were the phthalate metabolite MBZP and phenols 2,5-dichlorophenol, propylparaben, 2,4-dichlorophenol, benzophenone 3, and bisphenol S. And these findings were supported by our single pollutant models that identified the phthalate MBZP and the phenol 2,5-dichlorophenol as important chemical predictors of total T4. Again, in general, our mixture models identified important chemicals that are found in varied products like plastics, cosmetics, sunscreen, deodorizers, and pesticides. Our results also highlight how mixture methods can identify meaningful chemical contributors that would have been lost had we only focused on single chemical models. For example, propylparaben, seen here as number 3, was contributing as much as 2,5-dichlorophenol to the mixture association. However, it was not identified in single chemical models. Lastly, we showed that in women carrying males, an EDC mixture was negatively associated with TSH. And this was due to a mix of phthalates and phenols, such as 2,5-dichlorophenol, ethylparaben, bisphenol A, MEP, DINP, MCNP, and triclosan. And as I said before, these chemicals, many of them are used as antimicrobial agents, or they're found in plastics, perfumes, or deodorizers. However, in single pollutant models, only ethylparaben was negatively associated with TSH, whereas MBZP and DBP were positively associated with TSH. And these final results for our second objective highlight the strength of modeling chemicals as a mixture, because we may have missed important chemical contributors like 2,5-dichlorophenol, which is actually the primary contributor to the negative mixture association. Also, the relationship of MBZP and DBP are probably not important when considering the actual exposure profiles, because although they are associated positively with TSH on their own, when we consider the whole mixture, the relationship is negative. Now that we have identified the overall relationship of the mixture with hormones and specific chemicals primarily responsible, we wanted to determine if there were nonlinear relationships of the EDC mixture with hormones. And that's because, as Diana had said earlier, both cell-based and animal-based studies have demonstrated relationships of EDCs with outcomes that do not follow linear trends. For this objective, we use Bayesian kernel machine regression, BKMR, as a reminder. And this figure up top shows the relationship of EDCs with sex steroid hormones progesterone, estradiol, and testosterone in all women. And these plots are dose-response curves of the relationship between the EDC mixture and each pregnancy hormone, where we can visually inspect for nonlinear relationships. However, in all women, we did not identify any nonlinear relationships for the sex steroid hormones. However, in women carrying females, we identified a nonlinear association of the EDC mixture with estradiol. Zooming in on that middle panel, we show that the EDC mixture was nonlinearly associated with estradiol in women carrying females. Specifically, if you look at this dose-response curve, you can see an inverted U-shaped relationship where the mixture is negatively associated with estradiol at both lower and higher levels of exposure. And this highlights the importance of investigating low-dose relationships and how, if only moderate levels of exposure are assessed, relationships could be missed. While this result is interesting, future experimental and animal studies will be needed to understand the exact mechanisms of how this relationship could occur. And lastly, although I said earlier our focus of using BKMR was not to identify bad actors, I think I would be leaving out important information if I didn't state that the inverted U-shaped relationship was due to phenols bisphenol A and propylparaben, which are both found in, bisphenol A is found in plastics, like Diana said earlier, and propylparaben is an antimicrobial paraben found in some cosmetics. Moving on to the thyroid hormones, here you can see free T4, total T4, and TSH in all women carrying females, and women carrying males. And using BKMR, we did not identify any nonlinear relationships. With those results in mind, I will discuss general conclusions, and Diana will address some important future directions. To summarize, we identified associations of the EDC mixture with altered sex steroid and thyroid hormones. Specifically, one of our major findings was that an EDC mixture was associated with markedly decreased levels of mid-pregnancy testosterone, and that relationship was primarily due to phenols, triclosan, and propylparaben that are found in personal care products. These findings provide insights into the hormone disrupting potential of certain EDCs within an exposure profile of actual women. We also identified a nonlinear relationship of the mixture with estradiol, highlighting the importance of studying nonlinear dose-response relationships in both experimental and epidemiologic studies. Finally, future studies should explore how these associations relate to pregnancy outcomes.
So thanks, Brad, for going through the study. So again, we have shown that the EDC mixture is associated with early to mid-pregnancy hormones, and it has been shown in prior studies that early to mid-pregnancy hormones are important determinants of pregnancy and birth outcomes. So what about associations of an EDC mixture with pregnancy and birth outcomes? Well, in our prior studies in iKids, we do have evidence that individual chemicals are associated with pregnancy and birth outcomes. So for example, in one of our studies published in Environmental Research a couple of years ago, we evaluated associations of parabens, again, which are antimicrobial agents predominantly found in personal care products and cosmetics. And these chemicals were measured exactly how Brad described earlier. And we evaluated associations of these chemicals with birth outcomes, specifically birth weight and body length. And overall, we found that methyl and propylparabens specifically were associated with smaller birth size. So lower birth weight and shorter body length. And these results were sex specific, where we only observed these results among female newborns. In another study published last year in Science of the Total Environment, we evaluated associations of the same 10 phthalates and replacements that Brad described, and we evaluated these chemicals as a mixture with gestational weight gain, which is an important determinant of pregnancy health and fetal growth. And overall, we found that the phthalate and replacement mixture was associated with lower weight gain. And again, this finding was sex-specific, where we only observed the results in women-carrying females. And in terms of the major chemical driver of the mixture association, we identified that major plasticizer phthalate, DEHP, was the one contributing the most to the association of the phthalate and replacement mixture with lower gestational weight gain in women-carrying females. Therefore, as a future step, it will be important to examine whether associations of parabens and phthalates with these pregnancy and birth outcomes still persist when including multiple chemicals in a single mixture, but also determine whether hormonal disruption may explain these associations. However, we also know that pregnant women are not exposed to just these chemicals that we discussed today, but hundreds of chemicals that are found in diet or personal care products and cosmetics, or also other related sources. Therefore, it will be important to further expand upon this EDC mixture to include additional reproductive and developmental toxicants that are also found in personal care products, cosmetics, and travel with the food supply. So for example, per and polyfluoroalkyl substances, PFAS, are one class of chemicals of recent interest. And PFAS are environmental contaminants that humans are exposed to predominantly via diet and drinking water. And PFAS were the focus of our most recent article, which was published 2 weeks ago in the International Journal of Hygiene and Environmental Health. PFAS are considered endocrine disrupting chemicals due to their thyroid disrupting effects, which have been demonstrated in non-pregnant humans and experimental animal models. However, the effect of PFAS on sex steroid hormones has not been as thoroughly investigated. Therefore, in our study, we evaluated associations of PFAS individually and as a mixture with 3 maternal sex steroid hormones, progesterone, testosterone, and estradiol. Both PFAS and sex steroid hormones were measured from the same blood sample collected at median 17 weeks gestation, as Brad described in detail. And so all the details of the study can be found in the manuscript if you're interested. But just to provide the highlights, overall, we found that PFAS individually were associated with altered maternal sex steroid hormones. Specifically, we found that PFOS was associated with higher progesterone and estradiol, and PFHXS was associated with higher testosterone. However, PFUDA and PFDEA were associated with lower testosterone. Interestingly, we found that PFAS as a mixture was not associated with sex steroid hormones, although we did observe a non-significant trend for a positive association between the PFAS mixture and estradiol. And so if you're curious more about this, again, the findings are included in the manuscript. And additionally, we found no consistent patterns when evaluating associations stratified by fetal sex. So it will be important to create a comprehensive mixture of chemicals to which pregnant individuals are exposed to determine how the pregnancy exposed them. So everything mom is exposed to during pregnancy impacts their hormones and ultimately the health, how that is related and explains and is important for the health of mom and baby.
Thanks, Diana, for providing some exciting future directions. Also, thanks everyone for listening so far. And with that, we would like to acknowledge the Strakowski group, the co-authors on the paper that we went through, our respective departments. In addition, I would like to thank MSU's DO-PhD program and the Institute for Integrative Toxicology, including the EITS T32 training grant for my support for the last 2 years. We would also like to acknowledge our other funding sources, as well as the iKids team of participants without whom none of this work would be possible. Again, here's the information from our paper and a QR code for the link. Thank you for listening, and we are happy to take any questions.
Wow, thank you. That was great. Okay, we are gonna do questions now, and I'm going to read the questions from the Q&A box. So we already have 5, so keep asking your questions and we will get to them. And I'm not going to call on which you guys decide which one of you wants to answer the questions. All right. So I'm just going to start with with the first, which was, and this was posted during, Brad, when you were talking about the methods, was maternal occupation also collected?
Yeah, it was. It just, in our method for covariate selection, it didn't end up getting included in terms of employment and education are usually fairly correlated. So usually one, like you only really need to include one, and education was the one we included in this study.
All right. Another question about the methods, was postpartum hypertension included in gestational hypertension criteria?
Maybe that might have been in the background. Like, so we specifically didn't evaluate gestational hypertension, but in terms of summarizing the studies looking at these chemicals with gestational hypertension, we focused more on what was happening during pregnancy, but there are definitely other studies focusing on mom's health beyond the pregnancy, including postpartum period and well after. And that's a very important point, and that's definitely more studies do need to focus on that.
And I know there's research on bisphenols and cardiovascular health in general, like separate, like not related to pregnancy, but like cardiovascular disease just at a population level. So it does make sense that it may be related to both hypertension, unrelated to pregnancy, but also related to pregnancy hypertension that may be more linked to like the hormonal changes that Diana discussed.
Okay, I just saw there's a related question, which you may have just answered and I missed it. Are there any associations with EDC mixtures and hypertension? Yes, right? Is that
I'm not sure if I haven't looked recently at the mixtures. I just know that there's studies that have looked at like bisphenols and replacements as single chemicals, but now I'm intrigued, so I'll probably look it up after.
Okay. All right. Can you talk about how often studies on chemical impacts on pregnancy health disaggregate data by the sex of the fetus? And if it's not often enough, can you say a bit more about what we may be missing?
So based on like the review of the literature, it doesn't seem like every study does stratify by sex. And so I think that's an important limitation, because we do know, at least in like what we're proposing, that hormones are the mechanism for a lot of the endocrine disrupting health effects, or health effects of endocrine disrupting chemicals. And so I think it's really important and potentially a missed opportunity to not look at results stratified by sex, because we know a lot of these birth outcomes like birth weight or neurodevelopmental outcomes, there are sex specific effects of endocrine disrupting chemicals on these outcomes. And so it's just important as like a base, I think, evaluate the overall association of the mixture of EDCs on health outcome or whatever outcome you're looking at. And then if there is that sex component, stratify by sex. I think that should just be part of everyone's analysis.
I agree.
All right. Is there truth in the fact that as endocrine disruptors cross the transplacental barrier during pregnancy?
Yeah, so there have been studies that have been able to detect like what I'm familiar with, phthalates, parabens in cord blood, and also in placental, and bisphenols in placental tissues. So that definitely does imply that these chemicals can cross the placenta, into the placenta.
Yeah, I think like older research, decades old research, thought that the placenta was a lot stronger of a barrier in terms of filtering out toxic exposures. But I would say like probably for the last like 20 plus years, it's been known that it's a lot leakier than people used to think. It's not as strong as like the blood-brain barrier. And so as Diana said, a lot of these chemicals do cross freely, and they're measured in amniotic fluid. They're measured in cord blood. So yeah.
All right, there's two questions relating to the testosterone. Does the lowering of testosterone affect the testicular development of boys, or is responsible for malformation of the sex organs? And Jerry Heindel suggests that since testosterone was affected, it might be helpful to measure AGD, which is genital distance, which can be measured at any age. So I don't know if there's follow-up with,
iKids does measure AGD. And so that's actually something that another grad student in our lab has explored AGD as an outcome.
We have
And
a
it's something
publication with maternal obesity and AGD by Maria Klobovis. Yeah, at the time, that was her name, if that's the study you want to look up.
And so it's definitely a future direction, or it's not even necessarily future direction of this work, but it is just it's something that we can explore in this group, is the hormonal determinants during pregnancy of AGD. And then in terms of testosterone and testicular development, I'm not exactly sure what measurements. I think iKids also assesses as the kids get older, like puberty markers of puberty. And so I think like Tanner staging takes into account testicular development. So that's something that as the kids in this cohort get older, someone will be able to explore hormone levels during pregnancy, exposure during pregnancy, and then outcomes like age at specific Tanner staging. And that would be really interesting, especially since animal studies have shown that these chemicals can result in changes in in the size of the testes and in hormone production. So that would be really interesting studies to follow up as that data is collected.
And it's also great that we have ECHO as well, because this can also be followed up in a large sample of pregnant women nationwide. So there's lots of opportunities to evaluate this.
Someone asked, was birth type outcome collected? Like, can you compare chemical levels to C-section, induction,
Yeah, so like I mentioned, we did have some birth outcome information. So the one study we did measures of birth size. So yeah, birth weight, body length. We did have gestational age at birth. Unfortunately, iKids, less than 5% of births are considered preterm. So it's not
Fortunately, not
yeah,
unfortunately.
I guess fortunately, yes, fortunately for the health, but unfortunately for us. Yeah, and in terms of, so those are all important questions. C-section, vaginal delivery, some of these we just haven't looked at. But it's definitely.
So in terms of iKids, most of the C-sections are elective C-sections because the population's so healthy. So we wouldn't have enough women to determine if any of these exposures are associated with like an emergency C-section.
Well, a spontaneous birth versus a non-spontaneous. Yeah.
Because I actually did look into it, and it's just the population's too healthy and the pregnancies are too like quote unquote normal deliveries.
Although we were able to publish a study, we were looking at diet and gestational age at birth, and we were able to kind of tease out what was spontaneous and what wasn't a spontaneous birth. And we did see that diet was still an important predictor of is an important like mitigator of earlier birth. It promotes like better diet promotes later birth, which is important. So we're still able to see these things in iKids, even though it's a pretty healthy sample of women once we kind of stratify those results out. But yeah, like all those birth outcomes are very important, and we'll need to follow up and kind of build upon this work.
Can you look at diet and the EDCs together in the same study?
Yeah, so the paraben paper actually that I presented, another aim of it was actually looking at whether a better diet quality could mitigate or attenuate the association between parabens and smaller birth size. And we ended up finding that in women with better diet qualities, we didn't see that negative association, but in women with worse diet qualities, that association of parabens with smaller birth size still remained in women carrying females. And so that's one example of where we've been able to kind of look at potentially an intervention of if people can't avoid being exposed to chemicals, can they do something else, like change the other aspects of their lifestyle to improve the health of mom and then baby. But then also there are other studies also trying to looking at all these EDCs and nutrients as like exposures all together with health outcomes. So there's a lot that people are doing that'll be really interesting with chemicals and diet.
Okay, two sort of related questions. Do you know if people are looking at occupational exposure to EDCs and, on pregnancy, and if people are looking at women of color and environmental justice communities? Do you know research on those?
I'm not as familiar with occupational exposure research. That's actually something that I would be interested in looking into, but I'm sure it exists. There are
Yeah,
definitely.
there's
Oh,
a
sorry.
lot of questionnaire, like here at least at UNC, like I've been noticing that a lot of like the pregnancy and birth cohorts here, they collect extensive occupational information, and we do as well. We just haven't looked at the data, like specifically, like how much, like what is like mom working part-time or full-time? What's mom's occupation, like occupational title, hours in contact with receipts, like that type of information. So it's being collected. It's just the extent of how it's being maybe looked at or published on, I think, or maybe a little unfamiliar with that. But
I guess I was thinking that maybe the question meant more like occupational exposure of like people working in like plants that are like canning products and like I don't know if that was.
well,
I
that's part
know,
of like with
yeah.
kids. I think we collected that, maybe that information.
Maybe like there are areas that have
High
like
contamination, yeah.
yeah, like plant workers. I'm sure there are studies that have looked at it. I haven't looked into it specifically. And then in terms of like women of color, there are lots of environmental exposure cohorts that are like that are exclusively recruiting women of color in order to identify or in order to look into these types of associations. The other nice thing is Diana said, and I talked about it in my part of the presentation, the ECHO cohort trying to create basically a nationally representative pregnancy cohort that you can look at, and that would be really powerful because you'll be able to kind of get away from the cohorts where they're a very niche type of woman that like we're hoping that we can generalize out to other populations. But with ECHO, we won't have to assume that we're being comparable. We'll actually be able to know as the cohort goes forward and we collect more information and run more studies.
All right. There's a couple more. We're over time, but I can keep going for another minute. Someone, it might be the same person. I'm going to combine these two. Can we guess that the use of cosmetics with endocrine disruptors directly affects the fetus? And as a future clinician, Brad, what would you recommend for pregnant people to reduce exposure?
So from the first question of directly affect, we know that these chemicals are from these products. What's difficult is identifying specifically, I guess, knowing for sure where they're coming from and what percentage, what amount of that chemical is coming from the product that you're identifying. In terms of what recommendations, I think one of the interesting things about like having that pregnant time window is that even though pregnant women are bombarded with like recommendations of eat this, don't eat this, do this, don't do this, at least it is a more like narrowly tailored window of time that you can try to address reducing levels of exposure. And so if we're trying to, if we're thinking that these are related to both pregnancy health and fetal health, that hopefully if we can reduce exposure during that time window, even if 3 years later the exposure goes back to normal, then maybe we will have helped. And so I would say more studies need to be done in terms of intervention studies to see like what types of interventions, either dietary or changing personal care product use actually results in lowered levels. For another paper I'm writing, I just read through, there's 4 review papers that came out all within the last like 2 years that review all of the intervention studies to try to reduce the levels of exposure. And the studies are very difficult to do. A lot of them have very few participants. They may only have like 12 people in them. And some of them have like paradoxical results where they tell people to reduce exposure and then they end up with higher levels of the chemical of like maybe a specific metabolite increases. which highlights the point that Diana brought up, which is regrettable substitution. So we have to be very careful that if we say, don't use this, we know what is being used. But an easy example, I guess, would be like replacing like plastic bottles with glass bottles if you can, or glass, because like we know that it's not going and getting a new water bottle that just has a different bisphenol in it that's not BPA. We hopefully know that glass is relatively better than the plastic water bottles. So that's kind of a long-winded answer, but more intervention studies, so then we'll be able to know what specifically we can target, and try to target the like more sensitive populations like women during pregnancy.
All right. Well, thank you. I think we're going to cut it here. So thanks very much for the talk. It was great. And I am going to pass it over to Haley to close us out.
Yeah, thank you so much. That was really, really great. So thank you so much, Sarah. And thank you, Brad and Diana, for your excellent presentation and for this important research. And thanks also for the great comments and questions from our participants. I'll wrap up now with some quick announcements. A video recording will be available on CHE's website soon, and you will all receive an email with a link to the video. And we encourage you to support the work of any and all of the groups involved in the EDC Strategies Partnership. You'll find links to each group on CHE's partnerships page. If you appreciate these webinars, please consider a tax-deductible donation to CHE and/or any of these partners.